Hello, and welcome to the first of probably 3 or 4 parts of Chapter 9, Cell Communication and Regulation of the Cell Cycle. So this lecture here will be part 1. It's just a quick introduction to the concept of cell communication and some of the mechanics thereof. So let's go ahead and get started. In cell communication, cells have to be able to talk to each other in order to sense change. We know a plasma membrane is, of course, essential for a cell, not just to house everything that makes up the cell and kind of protect it and isolate it, but it's also used for communication. And within a plasma membrane will be multiple types of macromolecules, proteins, usually with a sugar attached to them, another lipid, or even another protein. And they're embedded in that membrane. Some of them span all the way across the membrane. Some may just be attached to the inner surface or outer surface of the membrane. Either way, these different types of proteins are all used for some type of signaling. If a cell cannot determine what's going on in its exterior environment, it's not going to be able to adapt or change in order to survive in that environment. Just think about when it gets cold outside. If you walk outside and it's 30 degrees outside, you're not going to wear shorts and a t-shirt. You're going to go back inside your house. You're going to put on long pants, maybe a sweater or a jacket. You're going to adapt to the outside environment. Well, the same thing happens with cells. They have sensor systems, these proteins that are in their membrane, and they will sense what is changing or going on in their environment in order to respond appropriately. So in cell communication, cells will detect and respond to something that's going on in the environment. That's referred to as a signal. The signal, technically in biology, we call it a ligand. And the receptor is called a receptor. This type of communication is needed not just for sensing change and adapting to change, but it's also needed for coordination. We are multicellular organisms, and our cells all have to work together in conjunction in order for our entire body to work. So the coordination of cellular activities is necessary, and that can only occur through cellular communication. So cell communication, sometimes referred to as cell signaling, involves the incoming and outgoing of different types of signals. These signals are always received by some type of receptor. The signals themselves are what influence different properties of cells. So they can cause a cell to change its conformation. They can cause a cell to begin excreting a new compound or making a new protein. Can cause a cell to change the overall function of a cell. So what signaling molecule is received or even released sometimes is referred to again as a ligand, and the receptor is what actually receives that ligand. Now, different receptors will be specific for different ligands. Not every receptor is going to receive every cell signal out there. So why are they responding? Well, again, needing to change, needing to adapt to a changing environment. So what we have here is a picture of some yeast cells. And on the left is a yeast cell in an environment in which there's very little to no glucose. Now you can see here that this yeast cell does have receptors for glucose in its plasma membrane, right? So it's got these glucose receptors, and not a whole lot of them, but as glucose becomes available. So now we've changed the environment. Follow this arrow. We're over to the guy on the right. When we change the environment and we start adding glucose, these receptors will receive the glucose and send a signal into the cell telling the nucleus of the cell, hey, glucose is now available. We need to make transporters. We need to make a new protein that will allow us to bring glucose into the cell because we can use it for energy. So the DNA triggers the gene for the proteins that make up the glucose transporter. The glucose transporter is created, moves through all of the processing, and the Golgi apparatus tags it for inclusion into the plasma membrane. So now we have our yeast cell here who now has lots of glucose transporters. You can see glucose, follow this arrow, glucose is coming into the cell. As glucose comes into the cell, those genes in the DNA are also sending out the message to make proteins that are metabolic enzymes in order to break down the glucose and extract all that energy from all those carbon-carbon bonds. So this yeast cell is responding to the change of glucose concentration in its environment by increasing its ability to uptake glucose and metabolize glucose as an energy source. Of course, over here on the left, I just want you to go back to this original cell, and on the left here, notice, because there is no glucose, this cell is not spending any time or energy or resources in making those glucose transporters and glucose metabolic enzymes. There's no reason for it because glucose is not available. So if it just makes a couple of these glucose receptors, then when glucose becomes available, it now can divert some resources to making everything necessary for metabolizing glucose. But there's no reason to use up all of those resources if there's no glucose to metabolize. So it makes cells this ability to receive a signal and adapt to a changing environment, makes cells incredibly efficient with their resources. Here we have an example of phototropism. Now, phototropism occurs when you, if you've ever had a house plant and you put a house plant near a window but not directly in front of it, after a few days or weeks, the plant will start to bend and start to move towards the sunlight. And this is because it wants to maximize the amount of photosynthesis that it can carry out. So this response, this bending, is known as phototropism. In order for this process to occur, you can see here on the right, we've got phototropism occurring, these plants bending towards the sun. In order for this process to occur, these plants have to start responding to changes in light. And they do this by the production of a hormone known as auxin. Auxin is a signaling molecule, and it is transmitted from one cell to the other. So these cells make lots of auxin. And auxin causes plant cells to elongate, to get longer. Not necessarily thicker, but just longer. So you can see here in the bent blade of grass, you can see that these cells over here on the left are very, very long. Notice that this is the side of the plant stem or grass blade here that is not directly in the sunlight. Over on the right side, these cells do not produce auxin. The production of auxin is actually inhibited when light is by the degree of light that is on the plant. So the more light there is, the less auxin the cells are going to produce, the shorter the cells will be. So on the right side of this plant, we have very short cells, and on the left side, we have very long cells. And so this causes the plant to begin bending towards the sunlight in order to expose more of the cells to sunlight for photosynthesis. Remember, these are plants, and they want to carry out photosynthesis. Okay, so all of this is occurring up here in the growing region of this plant. So it's just another example of why a cell would need to respond to a signal. This plant is not getting enough sunlight, and it needs to expose more of its mesophyll tissue to sunlight for photosynthesis to occur. So it's going to start bending and carry out this process of phototropism. So how do cells relay signals between themselves? When cells are far away, when cells are close by, and some cells even will excrete a signal to signal themselves. So there are about 6 different ways that cells will relay signals. The first is called direct intracellular signaling. This is when we have 2 cells that are adjacent to each other and they're connected to each other by what's known as a cell junction. We're going to look at cell junctions in another lecture when we cover section 10.2. But cell junctions are where 2 cells are right next to each other and they are physically joined to each other. And they will literally pass a signal from one cell to the next, almost like a little hollow tube in between the 2 cells that they can pass a note through. The next is contact-dependent signaling. In contact-dependent signaling, one cell has molecules bound to its surface, and another cell has receptors that will receive that signal. So I have one cell with a signaling molecule stuck on its surface, and I have another cell with a receptor for that signaling molecule on its surface. And when these two cells come into contact with each other, they will signal each other. The cell with the receptor will be signaled. That's what was happening in the last lecture when we looked at that at the white blood cells that were rolling. The white blood cells have the signaling molecule, and the endothelial cells in the lumen of the venous tissue have the receptors. All right, autocrine signaling is when a cell begins to secrete a signaling molecule, but that molecule will bind to the cell itself as well as very close by neighboring cells of the same type. This is oftentimes used to increase cell density. Scar tissues, skin cells will do this when forming scar tissue, and also sometimes in repair mechanisms. Paracrine signaling is a signal that will only influence very specific cells that are targeted in a very close proximity. For example, the synapse that occurs in a nerve cell from one nerve cell to the next, it's called synaptic signaling. The cell that excretes the signal, and then the cell right next to it through a space called a synaptic cleft, that little space, the signaling will then be received. So it's a very specific signal. It is targeting a cell that is very nearby, as opposed to just being excreted and affecting multiple cells. Endocrine signaling, this is where we have very long distances, usually hormones, some of our nerve signals. This endocrine type signaling is when a cell excretes one signal and it travels through the bloodstream to another part of the body. These signals are usually very long lasting, and they stick around, and they are also often concentration dependent. So here's this picture is from your textbook, and it's just a visual example of the different types of signaling. So you can see we've got our direct intracellular through these little cell junctions, one cell to the next. We have contact dependent, so we have a target cell here with a receptor and another cell over here with the actual signaling molecule or ligand for this receptor. We have autocrine signaling, where this cell is excreting a molecule that goes out and is being received by both the cell excreting it as well as neighboring cells. When we get a fever, our hypothalamus does this. In paracrine signaling, we have a cell that is releasing some type of molecule, signaling molecule, and it's being received by neighboring cells. However, it is not being received by the cell that is excreting it itself. So it has one less step than autocrine. And then over here we have our endocrine signaling, where we have an endocrine cell over here excreting a signaling molecule that will travel through the bloodstream to some targeted cell very far away in the body. So in cell signaling, there are three stages. We first must activate the receptor. So we have receptor activation. This is when the ligand actually physically binds to the receptor. When ligands bind to a receptor, very seldom is it covalent bonding. Most of the time it is some form of non-covalent, like a ionic bond. Hydrogen bonds are very common, and even some van der Waals reactions. In signal transduction, this occurs once the ligand is bound to the signal. So the ligand's bound to the signal, and now the signal is going to be sent throughout the cell. This is usually done through a series of proteins. The receptor now becomes activated, and it'll stimulate some type of sequence of events within the cell. That sequence of events is referred to as the signal transduction pathway. We are going to study a few examples of signal transduction pathways that are used by the cell for multiple signaling purposes. And then we have, of course, how the cell responds. So once the pathway makes its way into the nucleus and activates some type of gene, we're going to see some type of response occur. These cellular responses can be a change in gene expression, like I just talked about. Sometimes it doesn't go to the DNA. Sometimes that signal may just go and alter the structure of some protein. And by altering the structure of a protein, we alter its function. Remember, proteins carry out most cellular processes, and they do so through their shape. Sometimes we can alter the activity of one or more enzymes. Enzymes are a class of proteins that carry out cellular processes and break things down or build things. They are referred to as biological catalysts. They make reactions happen within the cell that would not normally happen by themselves. So we can have these different types of responses just depending on what the cell's original purpose is. So here we just have an example of our signaling molecule, right? So here we have receptor activation. Our molecule is binding to this protein. Here's an unbound receptor here. You can, Here is an unbound receptor here, and you can see that it's unactivated, so this bottom end here is curled. But when the signaling molecule binds to it, it's a protein, so it changes shape. That change in shape causes activation of this red protein, which activates the purple protein, which activates the yellow protein. Think a long row of dominoes. This protein here can come over and target an enzyme. It can target a structural gene, right? Alter the shape of a cell or how a cell moves. If it does an enzyme, it can change its metabolism or some other cellular function. Initiate or stop a cell function. And then of course down here, this protein, a gene regulatory protein, oftentimes referred to as a transcription factor, will alter the expression, the genetic expression of the cell, which of course is going to change the function of the cell at that point in time. Now, all of this, particularly this receptor activation process here, is also referred to, we describe the relationship or how strongly a receptor binds to a ligand through what's known as a dissociation constant, or a KD. The KD is a mathematical description of how well a ligand binds to its receptor. How efficient is this process? And although the KD is usually figured out for most normal cellular processes, the KD is something that a pharmaceutical researcher or a doctor is really going to want to know about a drug. So many medications work by binding to cell receptors, by either binding the cell receptor and turning something on, or maybe turning something off. Sometimes we can bind to a cellular receptor, like say on the outside of it over here. So if we bound this receptor up here somewhere other than its binding site for its ligand, that's going to alter the shape of this binding site. If we alter this shape, then the signaling molecule cannot bind to the receptor. By binding out here, we don't activate the receptor, we just prevent the signaling molecule from being able to. So if we prevent the signaling molecule, we can now turn off this pathway with some type of medication. So although the dissociation constant, or KD, is describing the relationship between a ligand and a receptor, it can also be used to determine the efficacy or the efficiency of a medication or drug in its ability to either activate or deactivate different cellular receptors, which in turn is going to alter the function of cells. So in a KD constant, it is equal to the concentration of ligand times the concentration of receptor found on the cell. It is divided by the ligand receptor complex, meaning how many receptors are actually bound by the ligand. So if I increase the ligand concentration, I should in turn increase my ligand receptor complex. If I increase my receptor concentration, I should also increase my ligand receptor complex. However, these both have to kind of occur at the same time for my KD to go very high. So if half of the receptors of a cell are bound and are in complex, then the KD is going to equal the concentration of the ligand. So in this case, what we're trying to show you is that the concentration of ligand and receptor is related. They are kind of codependent on each other here. The KD values for most ligands and receptors are already calculated, but they sometimes are used to determine different concentrations in experiments. So what exactly is a ligand? I've been using the term, and the ligand is the signaling molecule itself. It binds to a receptor. It does so with really high specificity, meaning a single receptor is going to be specific for a single ligand, right? So receptor and ligand relationships are very, very unique. The binding is always going to be non-covalent because we have to allow this to leave, and covalent bonds tend to be much more permanent. The binding and release of a receptor and its ligand, it usually occurs very rapidly. It's very quickly. Sometimes that signaling molecule, the ligand, is altered, and sometimes it is not. When the ligand binds to the receptor, once bound, the conformation of the receptor is going to change because the receptor is a protein, and that's how these guys work. The receptor, however, does not usually cause a conformational change to the ligand unless it is an enzyme receptor. Enzyme receptors oftentimes are causing these ligand molecules to react with other molecules. In that case, the ligand now becomes what's known as a substrate. Here we have an example of a hormone receptor. This is a hormone receptor complex that's causing the cell to respond somehow. This over here is a ion channel. So we have these ligands here. This green ligand here is going to bind right here onto this protein. It'll cause this protein to open up, and then all of these little yellow dots will be able to pour into the cell. So there are three basic forms of cell surface receptors, and these are the three that we are going to look at. We're going to look at enzyme-linked receptors. The specific type are called RTKs, or receptor tyrosine kinases. We're going to look at G-protein coupled, or GPCRs, G-protein coupled receptors. And then lastly, we will look at the ligand-gated receptors. I'm going to end lecture 1 here, and we will pick up and cover enzyme-linked receptors, or RTKs, in the next lecture.